everyone, welcome back to my channel. And today we are going to solve one paper, which is the MCQ one or paper from 2025 May June. It's paper one with variant one. In the other videos, we are going to solve the past papers of the last 5 years. So stay tuned with the channel and let's see how we can improve this part, because this one is very critical. Students get confused in some of the questions and they usually make mistakes by getting confused. So let's solve it step by step so that we can easily solve it. Okay, first one is, which statement about light microscopes are correct? So this is the one that we usually use in our labs. We can plug in, but that is also a light. We are using a light source for the magnification. That's why it is called as light microscope. Let's go into the options. Let's see which options are correct or which are not. Number one, to calculate the magnification of a light microscope, the eyepiece lens and the objective lens magnifications are added together. So they're already multiplied. So they're added together is seems to be like incorrect. Number two, the resolution of light microscope is limited by the wavelength of light. Of course, because here we have light as a source of viewing things. So definitely it is going to affect the resolution, how clear the image is. So when we talk about magnification, magnification means how large the image is, and resolution means how clear is the images. So 2 is correct. Number 2, the division on the scale on the stage micrometer are closer together than the divisions on the scale of an eyepiece reticule. Okay, this can and this is incorrect. So the only correct option is C, that is 2 only. Number 2, a student measured the width of a mitochondria in an electron micrograph of an animal cell with the magnification of C. This one, the width was this, and what is the actual size? Let's just use the formula. The formula for magnification is that you might be very well aware that, sorry, magnification is equal to, we say it's like the size of image. I'll write AS, okay, for image, sorry, IS, not AS, for image size. Let me just erase it. Okay. So this one is the size of an image divided by the actual size. So let's just we have to find the actual width. So actual width will come over here. So the formula will become that actual width is equal to the image size divided by magnification. So it would be like, I'm doing it in opposite direction, so don't mind that. It would become, image size is 6 mm divided by 9600. So you can convert mm into like the micron meter as well. By multiplying it with 1000, it will become mm. So it will become 6000 divided by 9600. So the answer would be 0.6 micron meter. To convert this, let me just write it so that you're not going to forget. We'll multiply it with 1000. Clear? Next one is the photomicrograph shows a section of a stem of a plant. So it's a stem. So this one is a cortex. The here we have the vascular bundle, and this one is a pet. Okay. So let's go into the detail that what is Q. Number one, the cell wall contain pits. There are small. Can you see small spaces in between them? Yes, we can see. Then the cell wall contain cellulose and also lignin. Lignin would be present as it contains vascular bundle, and vascular bundle definitely contain lignin. It is connected to adjacent cell via plasmodesmator. It is not visual. It cannot be seen over here. So the correct answer could be 1 and 2. Simple. The next one is the diagram is taken from electron micrograph of a cell that secrete enzymes. Okay, when we talk about electron microscope, there are two type of electron microscope. One is the in which we see the details of you can say the very inside of the cell, or you can say organelles of the cell. But there is one at another type in which we can see actually how the process is happening. So if we look into the process, like how the process is happening, definitely scientists know how is this happening. So they're just asking you, where are the polypeptides for these enzymes made? Now these polypeptides are proteins. Basically, definitely different polypeptide makes a longer chain, which is protein. So it makes it is made from ribosome. Now just look at these position where a ribosome present. A is a vesicle, no, it doesn't contain ribosome. B is the smooth ER. Let me write for you. So smooth ER doesn't make protein because it doesn't contain the ribosome. This one is rough endoplasmic reticulum, rough ER. So yes, it can make proteins. And number D is the mitochondria. So the answer is C, which is rough endoplasmic reticulum, which contains ribosomes and make proteins or put amino acids in the sequence. Which comparison between a typical bacterial cell and a typical plant cell are correct? So they have given you certain options. Let's see which one is correct. Number one, the bacterial cell has a cytoplasmic DNA, while the plant cell have nuclear DNA. Okay, it is correct. Number two, in bacteria, no smooth ER is present, while in plant the ER is present. Yes. So this is one of the difference. Endoplasmic reticulum is present, but the smooth one is absent in bacteria. Number three, peptidoglycan cell wall found. Yes. And then the cellular cell wall found. Yes. And then the bacterial cell and plant cell, okay, it's oppositely written. The bacterial cell has 70s ribosome, while the plant cell has 80s ribosome. So this one is incorrect. So you have the correct option A as an answer. Clear? Then we have number six. It's a diagram of basically a virus. So virus has two main parts. One is the protein coat, and other one is the you can say the DNA or RNA. So there are other capsule may be present, or there are other things present. So this one is a capsule. It has antigens. Just take an example of coronavirus. So which row correctly identifies the labeled biological molecules? Let's just discuss it. What is X? Oh, sorry, WXYZ. So we say this W is can be RNA. Let's see if it's a double helix or if it's a single helix. So it seems like a double molecule. So we'll write it DNA. Sorry. Then the coat is made up of, let's say, phospholipid, this one. Then this protein code, oh sorry, I've already told you, this is a protein code. So we write protein, and these are, you can say, the antigens or some kind of protein which can be identified by other organism. So the correct option is B. Next one is the iodine test. The iodine test is used for starch, and the Benedict test is used for the reducing sugars, or you can say the simple sugars. So let's go into the detail of this question. The iodine test and the Benedict test were carried out on a sample of starch solution. So definitely iodine is going to give result, while this Benedict is not going to give result. So amylase was added to another sample of starch. Now here the starch is converted into reducing sugars. Here, let's say. So definitely it is going to show which contains amylase enzyme, because it is going to break down starch into the maltose or the smaller or reducing sugars. So then the Benedict test was carried out. Let's see, okay, which result is correct. Number one, iodine test before adding amylase. So the C means the negative result. So first of all, Before adding amylase, definitely the test would be positive. Before adding means starch is present. Before adding, let me explain here. Starch is present, so starch will show which result? Iodine. This means no amylase, means starch, and iodine test is positive. So it can be B or C. Benedict test before adding amylase, it would be negative. Definitely it would be negative. And Benedict test after adding amylase, it would be positive result. So the answer is C. Next one. Which row about alpha glucose and beta glucose are glucose molecules are correct? Number one. Okay, the main difference is about the arrangement of hydrogen and hydroxide group on the carbon number three. So let's say carbon atom on which the OH position is different and the cellulose contain both molecules. Let first discuss one. So the carbon on atom which the hydroxyl position is different, so it is not first, it is fourth. We have already discussed. So when we move from down, it would be fourth if it is not third. So it would be like C or D. Okay, then the cellulose contain both molecules. So in the cellulose, we have sorry, no position is present. So it would be this one. Okay, front cut this one. I'm doing eighth one again. Okay, which row about alpha glucose and beta glucose molecule is correct? Let's say carbon atom on which the hydroxide position is present. Okay, when we discuss alpha and beta glucose, the actually difference lies in this one. You can also watch the video of this biomolecule where you can find the difference between two. Okay, then the cellulose contain both molecules. No, cellulose contain only one type, so the answer is A. Which molecules are monosaccharide? Let's say the okay, ribose. Yes, it's a monosaccharide one. Okay, what is the difference between monosaccharide and disaccharide? So saccharide is the sugar unit, or we can say the carbohydrate unit, and monosaccharide mean one. So yes, ribose is a five ring, a single molecule or a basic molecule. The second one is the glucose. It's a six carbon one. Then we have deoxyribose. It is also a form of ribose. So yes, it is monosaccharide. While sucrose is not a monosaccharide, it's a disaccharide. So the answer is 1, 2, and 3A. The next one is many cells uses triglycerides for long-term energy storage. Okay, yes. Which term are correct additional function of triglycerides? Okay, these are part of fats of our body. Let's say okay, what they do. They provide buoyancy in some marine animals, so it is actually yes. There are the main components of cell membrane. No, the main component is actually they are part of cell membrane, but the main is phospholipid. So both phosphate group and the lipid group are playing important role. They're used in thermal insulation, definitely, if the layer is present. So the answer is 1 and 3 only. Sorry. Then we have 11th one. The diagram shows two different bonds. Okay, there is a bond between hydrogen and oxygen. Then there is a bond between the amine group and the carboxylic group. This is amine and this is carboxylic group. So this one is known as peptide. Okay, then there is an S and S sulfide linkage. So yes, sulfide linkage and peptide bonds are present in both. Let's say whether it's present or not. So this one is additional, which is called as hydrogen bonding, is also present in proteins. Let's say it's hydrogen bonding. So all three are present in proteins. Simple. Next one. What describes the quaternary structure of a collagen molecule? Means whatever we have done. So there would be a hydrogen bond, there would be sulfide linkage, there would be peptide bond. All three are present. So how they are arranged? So first thing is polypeptide that form alpha helix, which are held by the covalent bond. Let's say so there are many bonds. So peptides that form a double helix, which are held together by hydrogen bond. Not only hydrogen bond, but and another bond is also present. Four polypeptides that are held together by a covalent bond which form a helix. Okay, this one doesn't seems to be correct. Three peptides that form helix structure which are held together by hydrogen and covalent bond. Yes, this one is correct because there are sulfide linkages as well, and there are hydrogen bonds as well. The diagram shows the structure of the enzyme amylase. Okay, this is again a protein. It has a tertiary and the quaternary structure. Which level of protein structure result in the folding of amylase into its globular shape, means the final shape. So the final answer is the primary, secondary, tertiary, and quaternary. Before explaining this one, I have already told what these shapes look like. It looks like a tertiary one. So answer is C. Which statement explain why large volume of water maintain a constant temperature when an air temperature increases? So this is about the properties of water, that how it store heat. This is called as the one term which we use as the specific heat capacity. That's why it stores a lot of heat before raising the temperature by 1 degree. So it's this answer. So we'll discuss the next MCQs in the other video. You can switch to that.